I wanted to show you, I was trying to put some slides together, and I came across a presentation that I did back in 2020. That's 4 years ago now, believe it or not. And what I wanted to draw your attention to, let me just pick up the laser. No, that's the pen, sorry. Laser pointer, very good. That back in 2020, there are all these actionable tests that we do for patients with lung cancer, but then there were a lot of other mutations for which there was they were not targetable, there was no drug. But you know, today 3 of these mutations are now actionable. You know, the HER2 amplification, there have been trials, Destiny Lung trials with the antibody drug conjugate. There are PI3 kinase alpha inhibitors such as alpelisib, which are used primarily in the context of breast cancer. However, they are now being used also in the metastatic and recurrent settings for lung cancer. And also, everybody knows about the single actionable mutation of the KRAS that until now has been a non-actionable, very prevalent but non-actionable molecule that is driving cancer. Now this particular mutation has got tyrosine kinase inhibitor. So, you know, even in 4 years, there have been significant advances, and I'm pretty sure these lists are not comprehensive and they will continue to expand. Anyway, that was my little reflection on the point.
Yeah, so it all started with this trial of 50 patients with proven ROS1 fusion by both NGS and FISH. And these patients were, like we mentioned last time, very young patients. So this made a huge impact, given the very good response, up to 70% either complete or partial response, which you can see here in this waterfall on the left, with the 3 complete responses, and the green is partial responses, which were maintained for a median duration of 17.6 months, which was a huge amount of time. Yeah. And here in this diagram, we can see the impressive response with remission of adenopathies and liver lesions in a patient treated. So what is ROS1 gene? ROS1 is located on a long arm of chromosome 6 in a slightly unstable area of this genetic structure. So it's a highly preserved gene, but actually we only know the function related to the fertility in males, so the development of male genital tract. It's highly similar structurally with ALK, and then we can see that looking at the evolution of treatment, because crizotinib was inhibitor for both ALK and ROS.
And we were commenting on the fact that highly conserved in evolutionary terms, it means that it must be really important. Otherwise, why do you want to conserve it across all the species? And yet we cannot find really a very good reason for it. I mean, these are little pieces of nothing. And that translates in our inability to find good tissue controls for this antibody. Anyway, sorry, it's just, yeah.
All right, so in lung cancer, we are talking about 3 main categories of alterations. Obviously, with the immunohistochemistry and the, let's say, primary testing in newly diagnosed lung cancer, we are referring to the lung fusion or rearrangement. And this can be interchromosomal or intrachromosomal, which makes, in a way, testing difficult, especially with FISH.
Yeah.
And then, obviously, point mutations, but this appears with therapy. They're secondary to therapy, and they're actually point mutations that are specific to certain fusions. And then amplification, which is quite a common phenomenon in lung cancer, but it's very heterogeneous and is related usually to higher grade cancers.
Yeah.
And it's also a result of fusion.
Yes, probably these are fusion genes, so they're already essentially mutated genes that then they're amplified in the genome to try and produce more and more in the cell. Yeah.
Yeah, so this is, I think this was your initial,
Yes.
from your initial presentation, and all the table with the presumed or the expected frequency of ROS1 fusion in different types of cancers, and you've got the ridiculously high percentage in cholangiocarcinoma, but you also notice in other types of neoplasts that are not epithelial, like SP384 neoplast, 17%, which is high and is still valid today, this high percentage, and also inflammatory myofibroblastic tumor, which we know it has primary ALK alteration. But now this is a more recent kind of sequencing cohort, which was initially set up for patients that were have cancer or an unknown primary. But this shows that most carcinomas, basically all carcinomas in the general population, they don't have a higher than 2% frequency. Of course, there are subpopulations, but this is the general rule. This is a diagram, a more recent diagram to show the evolution of therapy, which is quite impressive, although we were, I think...
It is very impressive. I think from discovery to therapy in 7 years is unbelievable. Yeah.
Yeah. So...
Although I think, Adelina, you pointed out that actually the discovery was a little bit earlier than that, but in lung cancer, this is when it started to emerge. Yeah.
Yeah.
But you see this, you know, the approval of the treatment is 9 years later. This is incredible.
Yeah. So you can see how quickly after the first drug, then we developed drugs that cross the blood-brain barrier, and then these patients obviously required that they were treated with inhibitors of specific point mutations. So this is, we're out of the ROS fusion now. We have drugs that are efficient to resistant cases through point mutation. And this is just an overview of how we test for ROS1 fusion. So first we use immunohistochemistry as a screening test. So if IHC is negative, then we can sign out the case happy that there is no translocation. And then we can use FISH. And if we're not satisfied, or if there's high suspicion of ROS1 fusion, then we can do NGS, especially with RNA techniques.
Yeah. And it's just worth to mention the fact that there are a number of different antibodies available, and perhaps the early antibodies, which were not as sensitive and as specific, caused a little bit of uncertainty as to whether we could provide ROS1 with an upfront antibody test. And the latest antibody, so the spring antibody, is now of sufficient sensitivity and specificity to allow us to use these immunohistochemistry as a screening test, because we are confident we do not miss. So there are no false negative using this technology. But there are some false positives. So the initial screening needs confirmation. And I think Adelina was implying on the fact that yes, you can use this break-apart FISH. You know, you know the principle of break-apart FISH. So when the two genes are, the two ends of the gene are together, you see this hybrid color. And if they are translocated in different parts of the cells, the two colors are separated. So it's a break-apart FISH. And for a long time, this was the gold standard. But in fact, we know the limitations of break-apart FISH. It will not reveal translocations that are very close to each other. And also, it may reveal presence of translocations that are not coding. So again, the importance of immunohistochemistry is also quite key. And so, as Adelina was hinting, if you have very strong immunohistochemistry in a negative FISH, you may want to actually reflex to a third methodology, sequencing on some sort. Yeah, that's what's kind of.
And we get to my favorite part, because we're pathologists, I hope at least some of us online, we kind of want to, we take the tissue, we know we send it to a machine, and we feel a bit like in the air, not very sure of what's going on. So I just put this together. It's not very specific, but when to expect ROS1 fusion depending on the morphology. So in cases where the morphology is solid, you're more likely to have a ROS1 fusion. There have also been described some macro nucleoli, bigger nuclei with macronucleoli. The next one, signal ring features in solid clusters, also sometimes accompanied by bizarre cells. This is very specific and has a prediction of 100% for ROS1 fusion, although it's rare.
Very rare, yes.
And also mucinous carcinomas, mucinous adenocarcinomas that have some sort of more bizarre morphology. And of course, following therapy, you can have all sorts of cytological changes, like turning into a small cell morphology-wise, or a sarcomatoid appearance. So these are some examples of mucinous morphology or solid. And as I said, the odd ones are more likely to be ROS1 positive. So this had a more conventional morphology. So this was the only one that was ROS negative among these cases. And these are from our collection to show this kind of solid signet ring morphology. So you can see cells with eosinophilic cytoplasm that had this blue blob of mucin. And you can see the staining is a bit more pushed towards the periphery of the cell because of the mucus. This is another case with the same signet ring morphology. It's a bit crushed here, but you can also see the very ugly, bizarre cells admixed with it. This is a higher power of the same case. But also, this is just another adenocarcinoma and NOS on cytology specimen. And it doesn't need to have these particular features to be ROS1 positive. So you can see the more classic cytoplasmic staining of moderate to strong intensity. Another important point is the presence of co-mutations. So these are either a diagnosis or during progression. And this is where also one can notice, and one must recognize the importance of NGS, because if you have another mutation that is not very common during at the beginning of the disease, then you, with more, let's say, smaller panels, you can miss that mutation. So the most common are the EGFR, usually sensitizing mutation, exon 9 deletion, L858R, and exon 20 insertion. There are also KRAS and BRAF mutations. Extremely rare ALK fusion or c-MET alterations have been identified. So while when EGFR mutation is present, we know that the patient benefits from EGFR inhibition first. With the other co-mutation is a little bit more complex.
Yeah, but definitely these core mutations may signify escape from treatment. So that, yeah, so that there could be a reason for that.
This is another example with a case that progressed under ROS inhibitors, and it was a transformation more like towards small cell morphology, as you can see here, from more adeno to basically small cell.
There, yes.
And then this was associated with the loss of P53 and RB1, which were wild type or preserved initially. And
you think this is because of this situation is because of selection of clones that are more like small cells or have this morphology, or do you think there is a progression of the tumor itself and acquires, under stimulation acquires more mutations?
It's possible that because the cells are also morphologically different, so probably a selection of a group of cells that eventually proliferated and
if anybody is interested in this concept, they should read the work of Charlie Swanson, because he has a large amount of body of evidence about genetic evolution in tumors under treatment or without treatment. This is a very interesting subject. Yeah. So this is kind of a paper that describes the antibodies that we use, and as I say, more recent. So this particular one as a look at the cell signaling antibody, but also at the spring antibody, and I think now the spring antibody has become a better standard for us. And also it creates a structure about interpretation, which is actually quite good. It tells us what the staining intensity. I think I have a picture here of staining intensity. So for every pathology understand that, but this is good to see the actual picture. This is 3+ staining intensity, 2+ staining intensity, 1+ and no staining, and then you require a 2+ or 3+ staining intensity in a proportion of cells, and depending how sensitive and specific you want to be, you set your parameters. Now, in the paper, there are comparisons of the antibody, and this is a good comparison because the D4D6 was the clone that was used until relatively recently, and they seem to perform very similarly to the spring clone. You can see the staining intensities are very similar. But when you look at low-level expression, so this is the neighboring lung, you can see here, and maybe this is a high power, you can see here that this antibody picks up the lower level expression in the reactive cells, and here you are struggling to see the signal. And perhaps this slight difference in analytical performance will explain the fact that the SP384 is a better tool for us to use in this screening context. So when you look at this paper, you understand that it is possible to use the immunohistochemistry as a first-line screening test, with the understanding that if it's negative, you could be confident that there is no oncogenic fusion. Now, why would you want to do this if you just send everything for NGS? Who cares about this? And the reality is that most lung cancers that we diagnose and need to be treated, so these are the locally advanced and metastatic breast cancer that cannot be operated. They're diagnosed on an EBAS sample, on a transbronchial biopsy, on something that has very few cells. Now, one thing is, as a pathologist, to see the enemy in the eye and see that it's positive or negative. Another thing is that you send it for NGS and they all work on percentages, So, if you look very carefully in the data that comes out from NGS on the small diagnostic samples, the failure rate of NGS is significant, maybe 25%. That's a quarter of the patients where you have no data. So that's why if you do have tissue, it would be worthwhile to look at it with your tools, with immunohistochemistry and the microscope. Yeah. So if it's positive, there is a certain amount of false positivity, i.e. there is overexpression of the protein, but the protein is not fusion protein, and therefore it's not constitutively activated, and therefore it's not a driver. So you can block it, but it doesn't really has an impact, the same impact on the tumor. So second test that we kind of traditionally have always been using FISH, but now studies indicate that maybe if you have a positive immuno and a negative FISH, you should ask yourself, let's do sequencing just to make sure, because FISH fails as a failure rate as well. And that's the fourth point. Maybe you need to think about this. And what are the strategies for that? So unfortunately, fusion partners can be very varied. And the more sequencing people do, the longer the list of fusion partners. And of course, if you only look at the fusion partners you know, you will miss all the ones that you don't know. So the best way of doing this would be to get some message RNA, reverse transcribe it, and then sequence the cDNA, and then you can clearly see that it starts off as a ROS and it finishes as something else. So there you are. You have a translocation. So that would be probably the most robust thing you can possibly do. And for those of us that have no large investment in technology and resources and cannot afford the NGS, there are these little gadgets like the Adelina machine, which has a cartridge essentially for the translocations, where they do essentially this work and RT-PCR the system. And we're kind of waiting for this to be installed in the lab. And so hopefully next time we meet, we can tell you our experience of using these this Adelina for confirmation. Okay.
And now a little bit of extra something, because while we have an IHC test on slide that can be useful in many ways, we also use ROS1 in our lab actually as a diagnostic test, because we do a lot of derm path, and I'm sure quite many people do. And we use the fact that many spizoid lesions are driven by fusions, including ROS1, and this is quite prevalent. So about 17% of Spitz neoplasms, but also 25% of Spitz nevi. You might think is not a very difficult diagnosis with nevi, but there's so many situations in which
often you just don't even get the whole
we either
picture,
had a biopsy,
get a shave or something.
and we find that quite useful, but obviously within a panel to kind of get to the diagnosis of Spitz lineage. Of course, it can't make the difference between benign and malignant, but it's very helpful in difficult cases morphologically. So just want to share this example with you. A low power, the tumor is a bit pink, so it's kind of at this power, you can't really see it because of the collagen. So it's composed of these nests and of epithelioid cells, and towards, let's say, the deeper part of the portion, they look a bit more like fascicles, and they're composed of slightly spindly cells. And then when you look at higher power, in this case, let's say this is more convincing. Maybe that's not the most difficult example of Spitz, but it does stain with ROS1, so it's driven by ROS1 fusion.
So, and that's actually is interesting, because Adelina said this is not a difficult diagnosis. Well, these are very large cells, and many of us think, oh, could this be a melanoma? I mean, is this atypical enough? I don't know. But I think if
it's a bit more atypical than a classic Spitz.
And I think if you see that the tumor is driven by ROS and it's not driven by other things like BRAF V600E, etc., so that it gives you confidence that you understand the lineage of differentiation, and therefore your diagnosis becomes easier to reconcile. And I think these are splendid examples of antibodies that were born for molecular oncology, but actually given to the pathologist, then we find a way of using diagnostically. But of course, it will have a lot of sense. And this is not the only one. We use
ALK, which has been more widely used than ROS up until now.
Okay, so perhaps we thought you may like this kind of concept. I think this is another Spitz tumor. Again, you can see the expression of ROS1 in this.
This is a
neoplasm.
Low power diagnosis.
Cool. Very good. How are we doing for time? Very good. Bit of bibliography for